All right, get ready for it. It's everyone's favorite day. It's Punnett Square Day! All right, let's go back to the beginning though. Before we can do that, we have to pay a little homage to our favorite monk, Gregor Mendel. Okay, he was the father of modern genetics because he did lots of work with pea plants and did some amazing science back in the 1800s. And he was kind of the first one to have documented and traced different traits and characteristics and kind of figured out how some of this inheritance stuff actually worked. Okay. And so basically, he kept breeding pea plants with each other. And there's some vocab that goes on with this. So the parents would be the parental generation, the P generation, okay? And then their children are called F1 or filial one, okay? And then those children's children, kind of like the grandchildren, would be the F2 generation. Okay, and so what he did is he ended up looking at lots of different pea traits. He looked at flower color, seed color, if the seeds were round or wrinkly, pod color, pod texture, where the flowers were, if they were tall or short, things like that. So he was looking at all these different traits and watching how they changed from generation to generation and how they changed based off of who their parents were. Okay, and so here's what Mendel did. Is he took a purple flower and he bred it with a white flower, and all of the babies were purple, which is a little bit surprising because, like, wouldn't some of them be white, huh? And then he took some of these purple babies and bred them, or like allowed them to pollinate themselves, which in humans seems really weird, but for plants, don't worry about it, okay? And then in like the grandchildren generation and the F2 generation, we had mostly purple with some whites. And so Mendel was like, huh, this is really interesting. How does that happen? How does the white go away and then it comes back? So this made him think about a lot of things, right? And so basically what this means is that we've got kind of different versions of traits. Okay, and so for pea plants, it's flower color, purple versus white. Okay, and this science word here is for an allele. It's an alternate version of the same gene. Remember back that, so like here's a chromosome. A gene is just a segment of a chromosome that codes for a thing. Okay, and so this is the gene that codes for flower color, and this is the gene that codes for flower color. And there's a purple allele and a white allele. Okay. And basically the only difference between purple and white is the exact sequence of our DNA, our A's, T's, C's, and G's. You put them in one sequence, it becomes purple. You put them in a different sequence, it becomes white, okay? And so we are getting, obviously, copy here from a mom and dad. Remember, we learned about this last lecture. Okay, we're diploid or diploid. We got homologous chromosomes. We got two different copies. And this is really, really helpful because if you have one copy that doesn't, for some reason, is a little bit funky, at least hopefully you still have one functioning copy from your mom or your dad that you can still build like a flower from, let's say, okay? And so basically, obviously, in that first generation, all the whites went away. So what does that mean? That some traits might mask or cover up other traits, okay? So when you breed a purple and a white, it doesn't necessarily make light purple, it made all purple. And so the purple masked or covered up the white, okay? And so what's going on here is some alleles are called dominant. I'm sure you remember this from like intro bio. Another name for dominant is the wild type, okay? And what this means is it makes a functioning protein. Or you can have a recessive allele, in this case is the white, and so this is also called the mutant, and that kind of has a negative connotation, okay? Recessive doesn't always, and mutant doesn't always mean bad, okay, as we'll see later on. But what that means as far as the protein is it makes either a protein that doesn't work or it is not even present at all, okay? And so, again, dominant, recessive, wild type, mutant, they're synonyms. And so one thing, another flashback to some vocab would be genotype versus phenotype. Phenotype is what it looks like. Genotype is the actual genetic makeup. Okay, and so we're going to get into the difference with this in just a second. Remember that we can use letters, uppercase and lowercase letters, to designate the genotype. Okay, and so like if we're talking about purple and white here, we could use a big P and a little p. And so if you see the word true breeding, that means that it is homozygous. which means two of the same letter. So a true breeding purple would be two big P's, and so that's shown right here. And a true breeding white would be two little P's, so that's right here. So these were our parental pea plants that Mendel started out with. And after he crossed those and bred them, he had all the F1 generation was all purple. And we know from setting up a little Punnett square that all of the babies would be big P, little p. Okay, they'd be what we call heterozygotes. They have one of each. Okay. And just a flashback to Punnett squares. Remember, you would set up like mom or dad across the top. Doesn't matter which one. One goes across the top, one goes across the side. And then since I have a big B here and a big B here, I put them together in this square. Since I have a little B here and a big B here, I put them together in this square. And so that's how you set up a Punnett square. Obviously, this cross that we're seeing right here is not these babies, okay? Because all of these are all purple, okay? And this cross is a purple flower and a purple flower. So this would be our cross for the grandkid generation. So this is an F1 individual and F1 individual, and they ended up getting crossed. And so here are the babies produced, mostly purple with a couple whites, okay? Because these little bees were able to buddy up, and then our recessive genes were able to come together and be expressed, okay? So this is, again, the F2 generation Punnett square for that one. And we will do a quick Punnett square review in class if this is a little fuzzy, but usually this is one of the favorite things people do in Fit Bio, so it usually comes back pretty fast. Again, so homozygous means same, either both capital, both lowercase, and heterozygous means one of each, okay? And so if you have a homozygous true breed or homozygous, dominant, let's say, that would be two big P's. If you have a homozygous recessive, that would be two little P's, and heterozygous always be one of each. So in this last one, we saw that we had three different genotypes. You could be big B, or big P, let's say. You could be heterozygous, or you could be homozygous recessive, okay? Okay, so let's take a look at, for this last Punnett square that we just saw, what's going on with that. So you end up with one individual that ends up with two uppercase letters. We would say they're homozygous dominant, okay? And so that would be their genotype. We had two that were heterozygous, okay? One capital, one lowercase. And we had one that had two lowercase, and that would be called homozygous recessive. Okay, so notice we have one homozygous dominant, two heterozygous, and one homozygous recessive. So our genotype ratio would be 1 to 2 to 1. Okay, but remember, genotype is what the genes are, and phenotype is what they look like. Well, this makes a purple flower. These also make a purple flower. So with our phenotypes here, we end up with 3 purples and 1 white. So our phenotypic ratio is 3 to 1, which is different from our genotypic ratio because different gene combinations can make the same flower color, if that makes sense. Okay, so again, here's another graphic to put it side by side if this helps you, that the genotype and phenotype don't always match. We can have a 1 to 2 to 1 or a 25% to 50% to 25% for our genotypes, but our phenotypes end up being 3 to 1 or 75 to 25 with this particular match right here. So let's say we've got a purple flower. Now, I told you it was big P, little p, but like, how would you know that? How are you supposed to know that if you're just looking at the flower? How do you know if it's homozygous dominant or heterozygous? How can you determine it if you don't have like breeding records for flowers, which most people don't? Well, the way that you would do that is you would take that flower and you would breed it with the known genotype, which would be the homozygous recessive, because we know the only way to get a white flower is if you have little p, okay? And so what you do is you would take a purple flower, breed it with a white flower, and you look at the babies, okay? And so here's what you do. Purple and white, and you have 2 possible outcomes. If the purple flower was homozygous dominant, here would be your Punnett square, and all the kids would be purple. Or if your purple flower happened to be heterozygous, Here'd be your Punnett square. Here's the babies. You'd have half purple, half white. So this is how you can kind of work backwards to determine the genotype of a flower that is showing the dominant trait. And this is a way to figure out what its genotype was if it's unknown. If you have all of the dominant shows up as kids, you know that the parent was also dominant. But if you have some of the kids show up with that recessive phenotype, you know that the parent must have been heterozygous. So, Mendel followed this with all these different traits, and that's really great. But one thing you should know is Ps are really simple, and most things are controlled by one gene that has two alleles. But real life is never usually that simple, okay? And there are some other dominants and some other inheritance that you might remember. There's something called incomplete dominance. Remember we said purple flower plus white flower doesn't make light purple flower. Guess what? Sometimes it does. And that is incomplete dominance. So a classic example are like carnations and I think it's carnations. Anyway, with certain flowers, when you got a red and a white version, the pink is your heterozygotes. Okay. So unlike before, where this would be a purple, here this is an intermediary, a mix of the dominant and recessive. That's why it's incomplete. You see the two of them kind of mixed together in the heterozygotes. So here's an example showing you with our graphics from earlier. There's another type of dominance called codominance, okay? So here we've got a horse that has red fur and a horse that has white fur. In our flower example, if you had a red flower and a white flower, the babies were pink. Well, hopefully, there's no such thing as a pink horse, okay? So horses do something a little bit different. The babies here, the heterozygotes, they have fur that's like red and white speckled. There's both red fur and white fur on the horse. Okay, this ends up being called roan if you're not a horse person. And so both show up, and that's why it's codominance, because they both show up equally in the heterozygotes, if that makes sense. One great place you can see codominance in humans is with our blood type. If you remember back to the cell membrane and that we've got these glycoproteins, right, these surface proteins on our cells, and they mark as they're a marker to say if the cell belongs to us or not, right? Remember, the immune system can come around and touch and feel to see up if that's from me and not, and if it doesn't, it attacks. Remember that? Okay, so let me just break this down for you guys. On our blood cells, you've got different surface proteins, okay? And you can have type A glycoproteins, and you can have type B glycoproteins, or you can have no glycoproteins, okay? So if you have type A glycoproteins, let's say they're like in a little triangle shape. And if you have type B glycoproteins, let's say they're in a circle shape, okay? But because you've got like a copy from mom and a copy from dad, okay? If your mom has A blood and your dad has B blood, then you're going to have AB blood, and you're going to have both glycoproteins on your surface, which is kind of cool, okay? Or if there are no glycoproteins on your surface, that's what this would look like. And so when you hear someone has type A blood, that means they have type A surface markers on their blood cells. If someone has type B blood, they have B surface markers. If they have AB, they have both. And if they have O, then they have none, okay? So I hope this makes sense that if someone with A blood were to get a blood transfusion with someone with B blood, your immune system would attack the B blood because it would say, that's not from me. Does that make sense? Okay. And so when you're going to donate blood, trying to find compatible blood types ends up being really important so your immune system doesn't attack the donated blood. And so here's a little chart showing the different blood types and who can donate to who. And so basically O can donate to anybody because it has no surface proteins. And so that's really great, actually, which is why O blood is always in high demand. Also, you should know that a lot of the things that we think about as common traits are actually determined by many genes. That's called polygenic. So great examples would be skin color, height, weight, intelligence in humans. Okay, these things are not just determined by one gene. It's many genes coming together. And here's an example in dogs. It's called epistasis, where one gene affects another. So you don't have to memorize this, but let me just explain it to you. In dogs, their fur can have pigment or no pigment. Okay, so as it turns out, pigment is dominant to no pigment. If a dog has no pigment in retrievers, that means that their fur is yellow. That's what leads to yellow labs. Okay. If their fur has pigment, then that is going to lead to either a chocolate lab or a black lab. Okay. So either pigment, no pigment. That's the first thing. And then there's another gene that's the pigment concentration. All right. So if you have pigment, but a little concentration, that's going to lead to chocolate lab. If you have pigment, but a lot of concentration, that's going to lead to a black lab. Kind of cool. If you have no pigment and a little concentration, no pigment always means yellow lab, regardless. But if you have no pigment and a little concentration, the nose and eyes and lips are going to be brown. Fur is going to be yellow. If you have no pigment and a high concentration, then your fur is still going to be yellow. But instead of being brown around the nose, lips, and eyes, it's going to be black. Okay, so again, don't super stress, but just showing how sometimes like one gene can be influenced by another. Again, a lot of traits are not just as simple as big B, little b, but just wanting to show you some other things here. Lastly, we talked a little bit about this in our last lecture about how you get the gender from male, female. Now, female is 2 X chromosomes. And so when the mom produces an egg, all of the eggs have an X chromosome because that's the only thing that mom can give. Okay. A dad has an X and a Y chromosome. So half of the sperm end up with an X chromosome, half the sperm end up with a Y chromosome. And if we do the Punnett cross between a mom and a dad, we're seeing that, of course, there's a 50% chance of getting a female and a 50% chance of getting a male, which I'm sure you knew. But what a lot of people don't realize is, so then who determines the sex of the baby then? Which gamete, the sperm or the egg? Well, the egg can only have an X chromosome in it because moms only have X's. So actually the gender of the baby is determined by the dad. If an X sperm fertilizes the egg, congratulations, you have a girl. If a Y sperm fertilizes the egg, congratulations, you have a boy. And so that's where that comes from. Okay. And so here's just some recap of what we learned in this lecture. And as always, here's where we are, chapter 11 in the textbook.